Pathological and biochemical changes in apoptosis, cell shrinkage, cell size is reduced. The cytoplasm is dense and eosinophilic, and the organelles, although relatively normal, are more tightly packed. This contrasts with necrosis, in which an early feature is cell swelling and not cell shrinkage. Chromatin condensation, this is the most characteristic feature of apoptosis. The chromatin aggregates peripherally under the nuclear membrane into dense masses of various shapes and sizes. The nucleus itself may break up into two or more fragments. Formation of cytoplasmic blebs in apoptotic bodies. The apoptotic cell first shows an extensive surface membrane blebbing, which is followed by fragmentation of the dead cells into membrane-bound apoptotic bodies composed of cytoplasm and tightly packed organelles with or without nuclear fragments. Phagocytosis. Phagocytosis of apoptotic cells or cell bodies, usually by macrophages. The apoptotic bodies are rapidly ingested by phagocytes and degraded by the phagocytes' lysosomal enzymes. In HNE stained tissue, the morphological and the biochemical appearance के साथ है, the apoptotic cells appears as a round or oval mass of intensely eosinophilic cytoplasm with fragments of dense nuclear chromatin. Because of the cell shrinkage and the formation of apoptotic bodies are rapid and the pieces are quickly cleared by phagocytosis, considerable apoptosis may occur in tissue before it is apparent in histological sections. The absence of the inflammatory response can also make it difficult to detect apoptosis by light microscope. This is an apoptosis of an epidermal cell by immune reaction. The cell is reduced in size and contains brightly eosinophilic cytoplasm and a condensed nucleus. Mechanisms of apoptosis. Apoptosis results from the activation of enzymes called as caspases, so named because they are proteases containing a cysteine and active site and cleave proteins from after aspartic residues. Like many proteases, caspases exist as inactive coenzymes and must undergo enzymatic cleavage to become active. The presence of active caspases is therefore a marker for cells undergoing apoptosis. The process of apoptosis may be divided into an initiation phase during which some caspases become catalytically active and unleash a cascade of other caspases, and an execution phase during which the terminal caspases trigger cellular fragmentation. Regulation of these enzymes depends on a finely tuned balance between the abundance and activity of pro-apoptotic and anti-apoptotic proteins. Two distinct pathways converge on caspase activation: the mitochondrial pathway and the death receptor pathway. The mitochondrial intrinsic pathway of apoptosis. The mitochondrial pathway is responsible for apoptosis in most physiological and pathological conditions. It results from increased permeability of the mitochondrial outer membrane with consequent release of death-inducing pro-apoptotic molecules from the mitochondrial intermembrane space into the cytoplasm. Mitochondria are organelles that contain remarkable proteins such as cytochrome C, a double-edged sword that is essential for producing the energy, example ATP, that sustains cell viability, but that when released into the cytoplasm, an indication that the cell is not healthy, initiates the suicide program of the apoptosis. The intrinsic pathway of apoptosis continued. The release of pro-apoptotic proteins such as cytochrome C is determined by the integrity of the outer mitochondrial membrane, which is tightly controlled by the BCL2 family of proteins. There are more than 20 members of the BCL family, which can be divided into 3 groups based on their pro-apoptotic or anti-apoptotic function and the BCL2 homology, which domains they possess. BCL2, BCLXL, and MCL1 are the principal members of the anti-apoptotic group. They possess 4 BH domains called BH1 to 4. These proteins reside in the outer mitochondrial membrane as well as in the cytosol and the ER membranes. By keeping the mitochondrial outer membrane impermeable, they prevent leakage of cytochrome C and other death-inducing proteins into the cytosol. Proapoptotic, BAX and BAC are the two prototypic members of this group. They contain the first three BH domains, BH1 to 3. On activation, BAX and or BAC oligomerize within the outer mitochondrial membrane and enhance its permeability. They form a channel in the outer mitochondrial membrane that allows the cytochrome C leakage from the intermembranous space. Regulated apoptosis initiators, members of this group, including BAD, BIM, BID, PUMA, and NOSA, contain only one BH domain, the third of the four BH domains, and hence are sometimes called as BH3 only proteins. The activity of BH3 only proteins is modulated by sensors of cellular stress and damage. When upregulated and activated, they can initiate apoptosis. Growth factors and other survival signals stimulate the production of anti-apoptotic proteins such as BCL2, thus protecting the cells from apoptosis. When cells are deprived of survival signals, suffer DNA damage, or develop ER stress due to accumulation of misfolded proteins, BH3 only proteins are upregulated through increased transcription and or post-translational modifications. Example, phosphorylation. These BH3 only proteins in turn directly activate the two critical proapoptotic family members, BAX and BAC, which form oligomers that insert into the mitochondrial membrane and allow proteins from the inner mitochondrial membrane to leak out into the cytoplasm. BH3 only proteins may also bind to and block the function of BCL2 and BCLXL. At the same time, synthesis of BCL2 and BCLXL may decline because their transcription relies on survival signals. The net result of BAX-BAC activation, coupled with the loss of the protective functions of the anti-apoptotic BCL2 family members, is the release into the cytoplasm of several mitochondrial proteins, such as cytochrome C, that can activate the caspase cascade. Once released into the cytosol, cytochrome C binds to a protein called as APAF, apoptosis activating factor 1, forming a multimeric structure called the apoptosome. This complex binds to caspase 9, the critical initiator caspase of the mitochondrial pathway, and promotes its autocatalytic cleavage, generating catalytically active forms of the enzyme. Active caspase 9 then triggers a cascade of caspase activation by cleaving and thereby activating other procaspases such as caspase 3, which mediate the execution phase of the apoptosis. This is the viable cell compared to apoptotic cell. Viable में survival signals होते हैं, growth factors, anti-apoptotic factors, fruit का production होता रहता है और cytochrome C का leakage नहीं होता है. उल्टी तरफ lack of survival signals है, irradiation है, DNA damage है. इनसे क्या होता है कि activation होता है sensors का, BH3 only proteins, antagonism of BCL2, तो activation होता है BAX-BAC channel का, लीकेज होता है साइटोक्रोम सी और एक्टिवेशन हो जाता है कैस्पेज का, लीडिंग टू एपोप्टोसिस. Leading to apoptosis. The extrinsic death receptor initiating pathway of apoptosis. This pathway is initiated by engagement of plasma membrane death receptors. Death receptors are members of the tumor necrosis factor receptor family that contain a cytoplasmic domain involved in protein-to-protein interactions. This death domain is essential for delivering apoptotic signals. The best known death receptors are the type 1 TNF receptor, TNFR1, and a related protein called as FASC, CD95. This mechanism of apoptosis induced by these death receptors is well illustrated by FASC, a death receptor expressed on many cell types. The ligand for FASC is known as FASC ligand, FASL, This is expressed on T cells that recognize self-antigens and functions to eliminate self-recognized lymphocytes that also express the receptor FAS upon recognition of self-antigens, and on some CTLs that kill virus-infected and tumor cells. When FAS ligand binds to FAS, three or more molecules of FAS are brought together, and the cytoplasmic death domains from a binding site for an adaptive protein called FAD, FAS associated death domain. Once attached to this complex, FAD binds inactive caspase 8 or 10, bringing together multiple caspase molecules and leading to autocatalytic cleavage and generation of active caspase 8. In turn, active caspase 8 initiates the same executional caspase sequence as in the mitochondrial pathway. This extrinsic apoptosis pathway can be inhibited by a protein called FLIP, which binds to procaspase 8, thereby blocking FAD binding, but cannot activate the caspase. Some viruses and normal cells produce FLIP as a mechanism to protect themselves from FAS-mediated apoptosis. Now, ये हम देखो एक्सट्रिंसिक पाथवे देख रहे हैं, फास्ट लिगैंड से बाइंड होता है तो फैड या डेथ डोमेन एक्टिवेट होता है. इट एक्टिवेट्स प्रोकैस्पेज 8 को कैस्पेज 8 में एग्जीक्यूशनल कैस्पेजेस एंड फाइनली होता है एपोप्टोसिस. Execution phase की बात करते हैं. The intrinsic and extrinsic pathways converge to activate a caspase cascade that mediates the final phase of apoptosis. The intrinsic mitochondrial pathway activates the initial caspase 9, जबकि extrinsic activate करता है 8 और 10 को. These active form of these caspases trigger the rapid and sequential activation of the executional caspases, such as caspase 3 and 6, which then act on many cellular components. For instance, once activated, these caspases cleave an inhibitor of a DNase, making the DNase enzymatically active and allowing DNA degradation to commence. Also proteolyze structural components of the nuclear matrix and thus promote fragmentation of the nuclei. अभी removal of the dead cells कैसे होता है? The formation of apoptotic bodies breaks cells into bite-sized fragments that are edible for phagocytes. Apoptotic cells and their fragments also undergo several changes in their membranes that actively promote their phagocytosis, so they are most often cleared before they lose membrane integrity and release their cellular contents. In healthy cells, phosphatidylserine is present on the inner leaflet of the plasma membrane, but in apoptotic cells, these phospholipid flips out and is expressed on the outer layer of the membrane, where it is recognized by the several macrophage receptors. Cells that are dying by apoptosis also secrete soluble factors that recruit phagocytes, and macrophages themselves may produce proteins that bind to apoptotic cells, not the live cells, leading to their engulfment. Apoptotic bodies may also become coated with the natural antibodies and proteins of the complement system, notably C1q, which are recognized by phagocytes. Thus, numerous ligands induced on apoptotic cells serve as a eat me signals and are recognized by receptors on phagocytes that bind and engulf these cells. This process of apoptotic cell phagocytosis is called as eprocytosis. It is so efficient that the dead cells disappear, often within minutes, even without leaving a trace. In addition, production of pro-inflammatory cytokines is reduced in macrophages that have ingested apoptotic cells. Together with rapid clearance, this limits inflammatory reactions even in the phase of extensive apoptosis. ये पूरा एक कंप्लीट पाथवे है. माइटोकॉन्ड्रियल पाथवे दिख रहा है, डेथ रिसेप्टर, फास्ट फैट लाइगैंड, इनिशिएटर कैस्पेज. यहाँ पे वही माइटोकॉन्ड्रियल लीकेज हो रहा है, साइटोक्रोम C का, रेगुलेटर्स हैं, इनिशिएटर, एग्जीक्यूशन कैस्पेज. कुछ भी करके ब्लेब्स बन रहे हैं, ब्रेकडाउन हो रहा है, एपोप्टोटिक बॉडीज बन रहे हैं, यहाँ पे फेगोसाइटोसिस हो रहा है. We talk about autophagy. Autophagy is a process in which cell eats its own contents. It is evolutionarily conserved survival mechanisms whereby in states of nutrient deprivation, starved cells live by cannibalizing themselves and recycling the digested contents. Implicated in many physiological states, example aging and exercise, and pathological processes. It proceeds through several steps: nucleation and formation of an isolation membrane, also called a phagophore, formation of the vesicle, called the autophagosome, from the isolation membrane, inside which intracellular organelles and cytosolic structures are sequestered, maturation of the autophagosome by fusion with lysosomes to deliver digestive enzymes that degrade the content of the autophagosome. This is one diagram. These are the cytoplasmic organelles. Initiation and nucleation complex बनता है. Initiation कैसे हो रहा है? यहाँ पे उसको अंदर लिया, फिर ये धीरे धीरे elongate होके एक autophagosome बन रहा है. This gets fused with lysosomes. Lysosomes are fused with. The moment it is fused, hydrolysis release करेंगे lysosomes के and degradation हो जाएगा. This is autophagy. Now, intracellular accumulations. One of the manifestations of metabolic derangement in cells is the intracellular accumulation of the substance that may be harmless or cause further injury. In many cases, if the overload can be controlled or stopped, the accumulation is reversible. In inherited storage diseases, accumulation is progressive and may cause cellular injury, leading in some instances to the death of the tissue and the patient. There are four main mechanisms leading to abnormal intracellular accumulations: inadequate removal of a normal substance, secondary to defects in packaging and transport, as in fatty change in the liver, accumulation of endogenous substance as a result of genetic or acquired defects in its folding, packaging, transport, secretion, as with certain mutated forms of alpha-1 antitrypsin, failure to degrade a metabolite due to inherited enzyme deficiencies, typically lysosomal enzymes. The resulting disorders are known as lysosomal storage disease. Deposition and accumulation of an abnormal exogenous substance when the cell has neither the enzymatic machinery to degrade the substance nor the ability to transport it to other sites. Accumulation of carbon or silica particles is an example of this type of alteration. Lipid steatosis, fatty change. The term steatosis and fatty change describe abnormal accumulation of triglycerides within the parenchymal cells, often seen in liver because it is the major organ involved in fat metabolism, but also occurs in heart, muscle, and kidney. The causes of steatosis include toxins, protein malnutrition, diabetes mellitus, obesity, and anorexia. In higher income nations, the most common cause of significant fatty liver are alcohol abuse and NAFLD, non-alcoholic fatty liver disease, which is often associated with diabetes and obesity. Lipids, cholesterols, and cholesterol esters. Accumulation manifested histologically by intracellular vacuoles seen in atherosclerosis, xanthomas, cluster of foamy cells मिलते हैं इसमें, cholesterosis, gallbladder में, and Niemann-Pick disease type C. Proteins. Intracellular accumulations of proteins usually appear as rounded eosinophilic droplets, vacuoles, or aggregates in the cytoplasm. By electron microscopy, they are amorphous, fibrillar, or crystalline in appearance. Causes: reabsorption droplets हो सकते हैं या फिर defective intracellular transport and secretion of critical proteins जैसे कि emphysema. Accumulation of cytoskeletal proteins या फिर accumulation of aggregation of abnormal proteins. Hyaline change refers to alteration within cells or in extracellular space that gives a glassy, homogeneous pink appearance in routine histological sections stained with H&E. Is widely used as a descriptive histological term rather than a specific marker for cell injury. Variety of alterations में मिलता है: resorption droplets, Russell bodies, and alcoholic hyaline. In long-standing HTN and DM, hypertension, diabetes, the wall of arterioles becomes hyalinized, resulting from extravasated plasma protein and deposition of basement membrane material.